Hello and everyone, welcome to class 12 biology. In this video we are going to discuss about dihybrid cross and Mendel's third law of independent assortment. In my previous video I told you that Mendel has proposed three different laws: law of dominance, law of segregation and law of independent assortment. The first two laws he proposed based upon his result of monohybrid cross. His third law, that is law of independent assortment, it was proposed after his dihybrid cross. When he observed the results of his dihybrid cross, he proposed the third law, that is law of independent assortment. So let's see what is dihybrid cross and what is law of independent assortment one by one. So in a dihybrid cross, what we do is we make a cross between two individuals or two parental individuals which differs in two traits. In the monohybrid cross, remember, we made a cross between two parental individuals which differs in only one character. In dihybrid cross, we will make a cross between two parental individuals which differs in two traits or two characters. So let's take an example. So over here we have taken the pea plants, two pea plants, which differs in two traits. So what are the two traits? You can observe over here. The two traits under observation are the seed shape and the seed color. Look at the seed shape. The seed shape can be either round or it can be wrinkled. Look at the seed color. It can either be yellow or it can be green. And in the chart which is given in your textbook regarding the characters which Mendel chose to observe regarding the pea plants, we know that the round seed shape is dominant over the wrinkled seed shape. The yellow seed color is dominant over the green seed color. So over here we have two parents which differs in two characters. One of the parent it produces round yellow seeds and the other parent it produces wrinkled green seeds. So both of these parents, they are true breeding. So when a cross is made between these two, Mendel observed that all the F1 generation plants were round and yellow seed producing plants. So how did he go explaining this observation or his result? First he has to write down the genotypes. So the genotypes for these two parental individuals were, for this particular plant, the roundness of the seed is governed by two homozygous dominant alleles, and the yellowness of the seeds or the yellow color seed trait was controlled by two homozygous dominant alleles. So this is a true breeding plant and it is homozygous for both the traits. This is also homozygous for both the traits, two small r's and two small y's. Now this is very important. What kind of gamete will these two parents produce, right? So since they both are homozygous, homozygous for both the traits, these two parents will produce only one type of gamete each. Look over here. So this particular parent, which is round and yellow seed, which is producing round and yellow seed, it will produce gametes which has what? One capital R and one capital Y. And this particular parent plant, which is producing wrinkled and green seeds, it will also produce only one type of gamete, that is having one small r and one small y. Now, important thing to notice over here is that the gamete, it will take one allele from each pair. It has to take one allele from each pair. Logically speaking, the gamete has to take one allele from each pair. From these two, it has taken one capital R. From these two, it has taken one capital Y. So we cannot have a gamete which will have two R's or two Y's, because it has to have what? One allele for each trait, right? And this is also having one allele for each trait. So when these two gametes, they fuse during fertilization, F1 generation will be the result, and all the F1 generations were with round and yellow seeds, and its genotype will be heterozygous for both the traits. It will get one capital R from this parent through this gamete, and it will get one small r through this gamete from this particular parent. And similarly with the two Ys, one capital Y from this parent and one small y from this parent. So this is heterozygous for both the traits, and that is why it is called as a dihybrid plant. Now, if you remember, whenever Mendel carried out a cross, let it be monohybrid cross or dihybrid cross, he made sure that he self-pollinated the F1 generation to get the F3 generation. So, upon self-pollination of the F1 generation, what kind of result he got, we will see over here. The F1 generation. The F1 generation, when it was self-pollinated, it has to produce gametes, right? So how many different kinds of gametes or how many different types of gametes it will produce? So here it will get a little bit tricky. You have to take out your pen and paper and practice it on a piece of paper by yourself, because this thing, it may look simple, but I have observed that many of the students, they make mistake while forming the gametes of F1 generation. So how will the gamete form? This capital R can come along with this capital Y to form this particular gamete over here, one capital R and one capital Y. It is also possible that this capital R will also come along with this small y and form this particular gamete. Likewise, this small r can come along with this capital Y to form this gamete, and this small r can also come along with this small y to form the fourth type of gamete. So these are the four possible allelic combinations in the gametes, and therefore it will form four different types of gametes by each individual. So when we make a cross or make a cross between two F1 individuals, or we can self-pollinate the F1 individuals, we will see that we will get F2 generation. And in order to form the F2 generation, we have to use Punnett square. Punnett square is really useful in this particular case. We have to use Punnett square for monohybrid cross as well, but it is very important for you to use the Punnett square for the dihybrid cross. So on this particular plane over here, we will place the pollens, the first set of gametes from the male parent, and over here we will put the gametes from the female parent, and these are the eggs. So when this and this gamete fertilizes, right, this gamete can fertilize with this one, there is a possibility. So what will be the result? So result we have to write down in this box of the table. So it will be one capital R from this parent and one capital R from this parent, right? One capital Y from this parent and one capital Y from this particular parent, and the resulting will be homozygous for both the traits. What about this one? This particular gamete can fertilize with this one as well. This pollen can fertilize this particular egg. There is a possibility. So what will be the result? 2 capital R and 1 capital Y and 1 small y. Now here you have to remember that the small letter or the recessive allele has to be always written after the dominant allele. You cannot write small y and a capital Y over here. It is by convention. You may do that, but it is advised, it is more convenient for you to write down in this particular manner. And these two can fertilize to give this particular F1 generation. These two can fertilize each other and give this particular F1 generation. And likewise, you can fill all the other boxes, right? You need to practice this. Just remember that you need to practice this. It may look simple just by looking at the slide or just by looking at your teacher writing down on the board. It may look simple, but when you go and write down on a piece of paper without practice, it will be very difficult. So you have to practice on a piece of paper. So these will be the results. So these are all the possible phenotypes of the F1 generation. These are all the possible F1 generation genotypes. I'm sorry. These are all genotypes. So now let's look at their phenotypes one by one. So this one will be round and yellow because both the alleles are dominant. These two alleles will code for round seed shape, and these two alleles will code for yellow seed color. So what about the next one? This will also be round and yellow, but here the second one is heterozygous, but still it will be yellow because Y is dominant over the small y. And similarly, all other round yellow we can find out. So this is also round yellow, this is also round yellow. So all these are round yellow. In total, we can count 1, 2, 3, 4, 5, 6, 7, 8, and 9. So we have 9 round and yellow seed producing F2 generation plants. So now, what about the remaining blank ones? So this, if you look over here, we have got 2 capital R's. So obviously the seed will be round in shape. But what about the seed color? The seed color will be green because these are 2 recessive alleles over here. So the seed color will be green. So what we have is a round and green seed. And look, you can find out other round and green seed producing F2 generation plants. So this is also round and green, and this is also round and green. So we have got 1, 2, and 3 F2 generation plants which produces round and green seed. What about the remaining 4 now? Let us look at this one. So this is having 2 recessive alleles for the seed shape. So the seed shape will be wrinkled. But it is having 2 capital Y's or 2 dominant alleles for the seed color. So the seed color will be yellow. So it will be round, wrinkled yellow. Okay, so this is also wrinkled yellow and this is also wrinkled yellow because this one is having capital Y or this one is also having capital Y. So the seed color will be yellow, but the seed shape will be wrinkled. Okay, so over here. And the last one, it is homozygous recessive for both the traits, and therefore the last one will be wrinkled green. Okay, so this will be wrinkled green. Now, what are the phenotypic ratio of the F2 generation? So all these boxes, they represent the genotype and the phenotype of F2 generation plants. So the F2 generation plants, they have got different phenotypes, right? So we have to find out the phenotypic ratio. Let us look at this one. So we have got round and yellow, right? Round and yellow seed shape and color. We have got round and green, and then we have wrinkled and yellow, and finally we have wrinkled green. So what are the proportions, or what is the ratio of these characters? So I already counted. We have got round and yellow 9 individuals. We have got round and green 3 individuals out of 16. And we have wrinkled and yellow 3 out of 16, and finally we have wrinkled green 1 out of 16. So what will be the phenotypic ratio? That will be 9:3:3:1. I have written over here 9 round and yellow, 3 round and green, 3 wrinkled yellow, and 1 wrinkled green. So that is 9:3:3:1. So this is the Mendelian dihybrid ratio or dihybrid phenotypic ratio. Okay, so in your textbook they have also asked you how many different types of genotypes are there. How many different kinds of genotypes are there in the F2 generation in a dihybrid cross? That you have to find out on your own. You have to search for answer through internet, or you can count how many different types or how many different kinds of genotypes are there in F2 generation. So for practice purpose, I have given you this particular cross. You have to do it just like an assignment. If you were in class, I would have asked you to do it in the class itself. For practice, I want you to make a cross between these 2 individual plants. One plant which is tall and purple. One plant which is tall and produces purple flower, and another plant which is dwarf and produces white flower. For your convenience, I have given you the genotypes over here. Now you should make a cross between these 2 individuals, get F1 generation, do the self-pollination for F1 generation, and produces F2 generation by using Punnett square. So you have to practice by using these letters, right? So I'm asking you to practice because I have told you it may look easy while you are looking at the screen or while you are looking at the board while teacher is explaining. However, when you go and write down in a piece of paper, it will prove a little bit tough. It will prove a little bit tricky. So with proper practice, it will come easy for you. Now, by looking at the dihybrid cross, I told you Mendel proposed the law of independent assortment. So what does a law of independent assortment state? The law of independent assortment states that when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. What does it mean? Right? You have to memorize that. You have to remember this particular statement. I will explain you the meaning of this particular statement. When the two pairs of traits are combined in a hybrid, right? When in our example, we had a pair of traits for the seed shape, that is round and wrinkled, and we had a pair of traits for seed color, that is yellow and green. So when they are combined in a dihybrid cross, we see that the segregation or the separation of the pairs, they are independent, or they are not dependent upon each other. It is not necessary that the round seed shape should always come along with yellow seed color. It is not always necessary. If we go back, we can see over here that the round seed shape, it is not necessary that it should always come along with yellow seed color, because the round seed shape, it can come along with green seed color as well. Likewise, it is not always necessary that the wrinkled seed shape should always come along with green seed color, because we have the example over here. We have wrinkled seed shape along with yellow seed color in the F2 generation. That means the traits, they have segregated, or they have segregated independently of each other. So that is the statement of law of independent assortment. Another statement that you can find in the internet or through Google is the law of independent assortment states that genes for the different traits assort independently of each other during gamete formation. So during gamete formation, what happens? The alleles or the genes for the different traits, they assort or they segregate. While segregating, they assort independent of each other during gamete formation. I have given you the diagram. This is the same diagram that we have seen before. So in this one, you can see that the genes, they are assorting independently. This capital R can go along with capital Y. The same capital R can also go along with small y. This small r can go along with the capital Y, and this small r can also come along with small y. So this is what the law of independent assortment states. And if you have studied about meiosis in your class 11, you may recall how the homologous chromosomes, they separate during anaphase 1. During the reductional division of the meiosis, how the homologous chromosomes, they separate. And the arrangement of the homologous chromosomes at the metaphasic plate, or the metaphase 1, is random. It is very random, so therefore it can segregate, or it can separate in random manner. So that particular concept will explain this particular law in a better way. So we will look into that one in our next videos. So for the next video, we are going to study about the chromosomal theory of inheritance. In the next video, we are going to study about the chromosomal theory of inheritance. So you all know what you should do. You should go and read about the chromosomal theory of inheritance before you watch the next video. Thank you.